Thank you, Melissa, for that lovely introduction. I appreciate it. First, I want to start this talk by helping you recognize that a couple of things. First of all, it's a highly collaborative body of work I'm going to be talking about, spanning faculty from Texas A&M University, lots of faculty at UT, and lots of both former and current undergraduates at the University of Tampa. The other thing I want to let you know is that I don't have graduate students. University of Tampa doesn't have a graduate program, so all the work that I'm going to be talking about is work that I've done with undergraduates. So it kind of sets the bar for the body of work that we've covered and the teaching tool that I've been able to use this research to engage in working with that population of students. So the work that I'm going to be talking about today, I think my title will be clear as we move forward. So one species, two species, hybrid species, new species. This is a discovery project. We started, I have been working with seahorses and pipefish now since my graduate work started in 1990. And so this has been a long history for me, and I started all of my work working in fish tanks in a lab up in Boston as part of my Tufts University background. And that was great work. It was a wonderful opportunity to work in fish tanks with these animals. But when you work in that kind of captive environment and you're interested in their behavior and the selection process on their populations over time, it's difficult to address those questions in captivity. You need to find wild populations to be able to study. And so in 2001, I took the position here at UT, thinking that it was going to be an easy transition. I would just show up in Tampa Bay, I'd put on a scuba tank, roll underwater, and there would be the seahorses just waiting for me to study their behavior. The species I started my PhD on are dwarf seahorses, which are about as big as your finger. So that's one problem. And seagrass beds are so complex that finding these tiny wee beasts in seagrass beds to study is difficult. Other things that were challenges as part of this work is that the water quality in Tampa Bay, although it's improved dramatically, it still is not as clear as it is in Caribbean waters. So I took a left turn and focused on not the behavioral ecology of seahorses underwater, but the population biology of seahorses in Tampa Bay, because it was difficult to study and ask the kinds of questions I was interested in asking. But that process changed, because in 2013, I have been working in the Bahamas for years now, doing reef survey and biodiversity surveys as part of different projects, looking always for seahorses and all of that work. Had found two in 14 years of work in the Bahamas. And in 2013, I got a phone call from a friend who said, I heard about these seahorses in a lake. And I was like, that sounds like a great story. So we're talking about these mythical, magical beasts, and somebody told me they found them in a lake. Not supposed to be, that's not where seahorses are found. They're not found in lakes. And so I took his word for it, got on a plane, showed up on the island of Eleuthera that I'm going to be talking about, and there is actually a lake full of seahorses. And so that's the story I'm going to be talking about today. So why did I start the work with this particular family? Well, I started the work with this particular family because I was interested in sex roles. I heard a couple of people when I was announced that I went to Smith College. Yes, that's a women's college, and yes, that was one of my interests. So to study this particular population, when I got to graduate school, I thought, if I'm interested in sex roles, we need to figure out how to ask good questions. And often, the way that we answer questions that we're interested in, it isn't using the traditional mating systems, the traditional organisms, to actually address those questions. Frequently, when we're trying to answer a question, we look for organisms that do things differently. And as many of you, I'm sure, know, seahorses do things differently. And so they're part of the family Cygnathidae, that includes seahorses, pipefish, and sea dragons. And the neat thing about this family is they have a range of specialization for paternal care, so dads care for the babies across the entire family. And so we have a range of specialization, which means that, for example, this pipefish here, although it's hard to see, there's bright orange eggs glued on the belly of this male pipefish. And so this is the simplest form of pregnancy in males in pipefish, where the eggs are simply glued on the belly. They develop there till their birth as independent offspring. Other pipefish species, like this Gulf pipefish here, they have brood flaps that enclose their embryos during development. So more specialization from there. Then we have the seahorses, and seahorses are one of the most highly adapted organisms for paternal care that we know of, because seahorses have these well-developed brood pouches in which their offspring develop. And I'll talk more about that. We'll take a peek inside the pouches in just a couple of minutes. The other organisms in this family also engage in paternal care. Our sea dragons, which are some of the most charismatic and amazing organisms within this family, brood their offspring in a brood patch, so open, similar to this pipefish in terms of their parental style. And pipe horses have a modified pouch, so they sit somewhere in the middle. And as you can see, the name pipe horse means something in between a pipefish and a seahorse. That long linear body of the pipefish and the crooked body of the seahorse is where the pipe horses sit. So the other thing, other than the parental care patterns that we see within this family, is that across this family we also see a huge range in their mating systems. And as a behavioral ecologist, that offers some really interesting opportunities to study how males care for offspring might have co-evolved with their mating system and their mating structure. And the mating systems range from complete genetic monogamy, as we've seen in the seahorse species that have been studied, where a male and a female during a reproductive event will actually, all the offspring within a male's pouch belong to one mother. So that's what we call genetic monogamy. But pipefish have a range of options in terms of their mating systems, from complete what we would call promiscuity, where males and females are mating multiply, and then pipefish where females are transferring eggs to multiple males, but males are only gestating the eggs from one particular female. So we call that a polyandrous mating system. And so this diversity of parental styles and the diversity of mating systems and the diversity of habitats offers a great model system for studying and understanding how a sense of place in an environment and the selective forces that are acting in that environment will actually help to shape a mating system and parental care patterns in a particular organism. And so I do my work, I work on both pipefish and seahorses, but this is a talk about seahorses, so let's talk about what we know. First of all, the question I often get, as you can probably imagine, is you just must have gotten the genders wrong. How is it that you know that males are male and females are female? Well, the definition of male is small and motile gametes. They swim around. And the definition of female is a large and non-motile gamete. So we know that females transfer eggs, the large non-motile gamete, through an ovipositor into the pouch of males. And it's inside that pouch where those eggs are fertilized. So the small motile gametes fertilize them. And then those embryos develop inside the pouch through a 10 to 30-day gestation period in seahorses. And the variation in time depends on the species, and in addition, it depends on water temperature. So they'll take longer to gestate if the temperature is colder. As you can see in this picture, so these are embryo seahorses. What we can see here, there is an eye. For an example here, this is the back of the seahorse. These are heads of seahorses. There's another eye here. And what you can see is they actually burrow into the surface, the epithelium of the inside of that pouch tissue. And as they burrow, the male's tissue actually wraps around the embryos and develops a vascular relationship so that the male is exchanging gases for his offspring, so breathing for them, as well as removing waste products. And we know from pipefish, although we don't know for sure in seahorses, there is some feeding that happens for pipefish offspring as well. So it's possible that males are actually feeding their offspring. So from this kind of pouch development and offspring development, we also know that they display elaborate courtship and greeting rituals, some of the most civilized among the fish species, I think. So every day, a male and a female will come together and will interact with each other even when it's not time to transfer eggs. They call them the greeting rituals. So a male will come up to a female, he'll quiver, the female will come up, she'll quiver back, and they'll do this nice little exchange for a few minutes, probably exchanging information about how far along the male's pregnancy is and whether it's time to transfer eggs yet. We think it's a way for them to synchronize their reproductive cycles. And then on the day, early in the morning, the male will give birth, and later that same day, she will transfer him another set of eggs. And so I know the women in the audience are saying, that's a great system. And I would agree. So I already mentioned that they're monogamous and that they are genetically monogamous. So work done by one of my former undergraduates in the lab, who's now a first-year faculty member at the University of Tampa working directly with me, actually did the genetic work to look to see that, in fact, the dwarf seahorses that I started my work on, as well as a number of other species, are genetically monogamous. So males and females are contributing to single offspring, a single group of offspring, and there's not multiple mating. And we also know about seahorses where we find them. And unfortunately for seahorses, but fortunately for me who studies them, they're found in shallow coastal habitats around the world. What does that mean? They're found in seagrass beds predominantly, but also mangroves. They're found associated with corals and gorgonians. So there's anything basically they can wrap their tail around, including dock lines, which I'm sure some of you folks have probably seen them on docks and places like that. They'll hold on to them, and you got to watch those dock lines when you pull them up, because you frequently will come up with a string of seahorses on them because they really like rope to hold on to. But of course, there's a problem associated with the shallow coastal water ecosystem where they're found, and that problem is that we as humans mostly live along the coasts. And because of that, we are causing, habitat degradation. We've got a loss of our submerged aquatic vegetation, so our seagrasses, for example, and a decrease in water quality, which is leading to a loss in seahorses worldwide. The other thing that's important to know about threats to these particular organisms is, well, this. You may have seen this in your local craft stores. Craft stores sell a range of shells. They sell dried seahorses for use in decorations of all sorts. And when you go to the front desk, they'll tell you that, oh, those were seahorses that washed up on the beach. But that's not true. They actually go collect these animals, they dry them out on docks, and then they sell them for, we call those curios. They're also heavily used, and I took this picture in Honduras a number of years ago. They're also used in traditional medicines. So they're used throughout Southeast Asia, as well as a number of South American countries. They're used in traditional medications or traditional medicines. And as a result, there is actually a seahorse fishery. And their populations are under threat. They're actually listed on the Convention on International Trade in Endangered Species as an Appendix 2 species, which means that their international trade is regulated because it's a population it's a group of organisms we're actually very worried about because of this intense fishing pressure. So, why did I do the work in the Bahamas? Well, why not go work in the Bahamas, right? I mean, seriously. So, why did I do the work here? Well, we knew that 3 species were found in the Bahamas, or at least I knew about that. I'd only ever seen 2 before this work started, so I just had to rely on the textbooks that told me that they were there. So the dwarf seahorse is supposedly found in the Bahamas, the lined seahorse Hippocampus erectus, and then Hippocampus reedi, the slender seahorse, or also sometimes called the long snout seahorse. So this is Zostera here, this is reedi, a long snout, and this is an example of a Hippocampus erectus. So we knew that they were in this location. The other thing that's great about the Bahamas is it's obviously a very biologically interesting ecosystem, but their tradition of fishing methods have been actually target fisheries. And from the perspective of seahorse populations, one of the biggest threats other than traditional medicines and collection for the aquarium and curio trade is our fishing methods. And so if you're trawling for shrimp and seahorses are about the same size, and so therefore, and they're also found in the same habitats. So if you're collecting them in that fashion, what you'll find is that you'll get lots of seahorses in shrimp nets. But by the time we pull all the shrimp out of those nets, those animals are dead, and we've lost them now from populations. So shrimp trawling and general fisheries hit these populations very hard. The Bahamas doesn't have a traditional shrimp trawling fishery. It's mostly target fisheries in the Bahamas, so their populations have not been depressed. And one of the most difficult aspects of working with seahorses worldwide is we don't know what normal seahorse populations look like because they've been decimated so broadly by all of these challenges to their populations. So looking for a baseline that we can actually use to understand what normal seahorse populations looks like is really important. And also, well, there's the clear, wonderful water in the Bahamas where if you're studying their behavior, you can actually lay a meter or so away and watch them do whatever it is that they're going to do, and they're not bothered by it. You don't have to be this far because your visibility is so low. So the visibility is great. It's a good system to work on. And this is actually a seahorse picture I took. This was one of those 2 that I took on Andros Island in the Bahamas. And so my looks for seahorses across the range, so this is the Bahamian Island chain, just to give you a sense, this is New Providence, this is Nassau, the capital. I've been doing my work, so I've spent time looking on Andros, I've spent time looking on New Providence. I got lucky enough to be able to do a research trip down here to the Ragged Islands. I've worked in the Exuma Keys. All of that time, 2 seahorses. But I got that great phone call and was able to start my work on Eleuthera Island with this particular population. So, the location I'm talking about, and as the title of my talk suggests, the Sweetings Pond Seahorse. This is, and the funny thing about, so this is what it's called, Sweetings Pond, and I invited my former undergraduate, now, and then she was a late graduate student. I invited her to come work with the pond. It was spring break. She's like, oh, great. We're going to go work in a pond. We'll finish it in like 2 days, and then the rest of spring break, I can go sit on a beach in the Bahamas, right? Because it's called a pond. Well, she showed up, and that's the pond. It's 1 mile long by ½ mile wide, and not something we could cover in 2 days. And so she's not been unhappy with it since, but we don't get much beach time because we have a lot of work to do in this ecosystem. And so what you're looking at here, this is from the south end of the lake, this is from the north end of the lake, and this is actually looking offshore. If you climb up the ridge off of the side of the lake up here and go to this ridge, this is what you see, the ocean on the outside. And as you can see, although we do see seahorses here, the other probably second most common organism that we see, which is amazing and you just don't get to see these very often, are octopus. Octopus are very common in this particular system. This pond, Sweetings Pond, is it's a tidal lake. We call these ankyline ponds. Other types of ankyline ponds, many of you have probably heard of. Blue holes, for example. So blue holes are very common throughout the Bahamas, throughout the Caribbean, and other locations. And these are well-known sites of what we call endemism. So when species get into these blue holes or get into these ankyline ponds and become isolated from the outside ocean, they're great sites to actually watch evolution happen in progress. Because the animals get isolated, there'll be drift, there'll be differences in selection, and all of a sudden you have a new species. And so that's one of the great things about this particular system. We also, the neat thing about this system too, excuse me one second. My phone is off, but my alarm went off. So I'm just going to turn that off. Don't want to get distracted by the alarm continuing to go off that I can hear that you may not be able to hear. So the other thing that's neat about this pond is that for a very long time it has been very understudied. So I am the first research biologist that has published any work on the pond since the early to mid-1980s. And so there was early work done on it, which I'm going to talk about in a second. But the neat part of it, or it's kind of an interesting part of the story, is that there are very few human impacts in this pond. So most blue holes throughout the islands are not seen as particularly valuable real estate. They tend to be places where people throw trash, where people often use it as a natural pond source to dump game fish to be able to raise them in different contexts. So a lot of these have had very heavy human impacts. Sweetings Pond, strangely enough, has had a long-time story about a sea monster that lives in it, and I think the sea monster has actually helped protect this system from exploitation, because it's almost a pristine system, which is highly unusual to be able to see in this day and age. To see a system that's this close to so many humans that very few people get the privilege to actually get in and have explored. And so this is just a map to give you a sense of the study area. So this is the Bahamas map that I showed earlier. This is Eleuthera. So this is where we are in Eleuthera. So this is Sweetings Pond. If you've been on Eleuthera, the Queens Highway runs right through here. And this is a water body I'm going to talk about at the very end of the talk. And this water body here is one that is similar in structure to Sweetings Pond. Sweetings Pond, when I talk about isolated ponds, one of the key issues when we talk about this isolation for Sweetings Pond is how do we know it's isolated? Well, it's not completely isolated, but we've actually deployed tide stations throughout the long axis of the pond, in addition to outside, to be able to look at that tidal change. And there's a maximum tidal change inside the pond of about 7 centimeters, whereas outside the pond, that tidal change is a meter. And so we know that there is some water flow, but typically in blue holes that are connected to the ocean, you'll see what's called a boiling hole. Well, you'll actually see water during tide influx that'll actually come up in a very large spot that indicates a big connection to the ocean. Sweetings Pond doesn't have any of those, and based on the modeling data that is work that I'm not going to talk much about, but based on modeling data, we've determined that in the pond, it must be not a big hole that allows water in, but this limestone Swiss cheese, so very tiny connections to the ocean, and thus very little connection in terms of the ability of organisms to go in and out. So we're going to talk about that as we move forward. And this is just, again, so this is an arrow of when you look off of this headland, this is what you see. So you're actually seeing the ocean out there. Okay, so what did we know about Sweetings Pond when we got started? Well, we knew a few things. So Richard Aronson, who's now based at FIT, did his graduate work in this ecosystem in the early 1980s. And he wasn't focused on seahorses. He was actually looking at two major groups. He was looking at the octopus in the lake, Octopus briaris, and he was looking at a brittle star and studying their populations because it's amazing to see, 15 octopus when you're in the water for 2 hours. It's just not something you can do anywhere else that I know of in the world. And so his work in this system as a PhD students tend to look at a large number of questions when they're working. So he wasn't just focused on these. He actually looked at the fish population in the lake. And what he found back in the early 1980s is that there's an extremely low diversity of the fish in this particular ecosystem. So back then, he found that there were 17 species from 15 families, and most of the fish that he saw were very small. In addition, and when he did this, he also made comparisons between inside the pond and outside the pond. Outside the pond, in the ocean, there were 126 species from 50 families, down to the same depth we see in the pond. So the pond has many fewer organisms, and all of them were little. There are some large fish in the pond, but we believe that those were stocked in the early 1970s, and we don't see them very commonly. So we've seen some groupers, some snappers, but other than that, we see mostly what we think are native fish that actually were just transmitted here. Again, we don't know how, but somehow managed to make it into this lake. Our more recent studies are, anything in orange here is stuff that we have found. We've actually done work recently, again, doing the same fish surveys that Aronson did in the early 80s, and we've found 13 species in 12 families, indicating that there's been a potential species loss over the course of the intervening 35 years. In addition, what Aronson found was he looked and found that this is what he called a predator-free environment. How did he learn that? Well, he noticed that, and you can see in this picture, octopus are not out at night, which is the normal time for us to see them. They're out and active during the day. The same thing is true of the brittle stars in the system. Most of the time you see brittle stars if you're out snorkeling or diving at night. These, that looks like the entire ground on the surface, the bottom of the pond is moving during the day because there's so many brittle stars and they're so active during the day. Aronson got interested in this, and he started, well, let's just start with a count, and found that both octopus and brittle stars were orders of magnitude higher in terms of their population sizes than he saw outside the pond in the ocean. So right there, that indicated there was something different going on. He also was able to study their behavior, notice that this was gregarious, active behavior during the day, and so he said, well, I wonder if this is a genetic differences. Are these new species of these two organisms, or have there been, or is there something else going on? So he did a classic transplant experiment. He took brittle stars from inside the pond and brittle stars from out in the ocean, and he swapped them, thinking that if these were genetic differences, the ones from the pond would stay gregarious when he moved them in the ocean, and the ones out in the ocean would stay hiding and they'd stay active at night when he moved them. He didn't see that. He saw that instantly when he moved pond brittle stars to the ocean, that they became very cryptic. They hid, they were only active during the night, and when he took the ones from the ocean and put them in the pond, they were active during the day, so they showed a completely different behavior, indicating it's probably something in the water, namely a lack of predator cues, that's actually controlling this behavior. So that's how he demonstrated that it's a predator-free environment. What we did to determine if this was predator-free is we actually deployed what we call BRUV units, so baited remote underwater video units, and we deployed them at a number of locations. They're the little circles on this map, so 7 locations around the map, around the pond. This is what they look like. There's a big unit here, there's a float on top, there's a camera out here, and then at the very end, we put a little bit of chum, we put a little bit of food out there in this flow container, and we turn the camera on and we let it go to see if we see any evidence of predators. And after watching hundreds of hours of nothing, we also determined that there are no predators in the pond in this condition. We do, however, know that there is this population of grouper and snapper. They engage in very strange behavior for grouper and snapper. They actually school at the surface in large aggregations all the time, not just during spawning aggregations. But this is a whole other question to be addressed in the pond. The other thing that Aronson noted is he actually looked at the bottom of the pond to try to understand kind of its native context, what's going on in this particular system. And so what he determined in this system is that the pond bottom, when he was doing his work, was covered with Cladophora. And Cladophora is a fast-growing algae that's associated with high levels of nutrients, probably associated with the high levels of agriculture around the pond that existed in the 1980s. That agriculture has since decreased and is actually has been non-existent for a number of years around the pond edge. And we see a habitat much more like this with slower growing algal species, and we don't see Cladophora as often. So we believe that there's been a change in the water chemistry and the water quality as well. This is a bathymetry map that we've generated from the pond just to give you a sense of depth. Blue holes can have variable depths. A lot of these salt ponds in the Bahamas are also very shallow. Ours actually fits somewhere in the middle. So it's got a very shallow edge, but it's got a couple of deeper holes. We think that the deepest spots in the pond that we've mapped so far are about 14 meters, about 46 feet deep. So it's got some deep spots, but most of it is about 3 to 4 feet around the edge, and that's where we've done most of our work. Aronson also observed that there were seahorses in the pond back in his work, but he actually observed them very rarely. And as you'll see in my talk, that's also changed since the 1980s, and they're no longer rare in this ecosystem. But his observations posed a challenge for us, because he originally described these as long snout seahorses, Hippocampus reedi. And so we went into it assuming that that's what we were seeing. Even though we know that both species are found outside in the ocean in Eleuthera, he said that they were reedi, and so we assumed that they were when we got into the pond and started doing the work. We know that these two species are actually distinct. They're two distinct populations that cluster separately when we do genetic work, and they're part of 2 different colonization events in the Atlantic that are separated by 14 million years. So we know that they're separate species. However, that definition of species is somewhat nebulous, because when you take these 2 species and you put them in a fish tank together, they actually interbreed and produce offspring. They produce hybrids. And so there's possibilities here. The animals in the pond could be reedi, they could be erectus, they could be a hybrid, or they could be a new species. And so we had to set out to actually address this question. There's no specific data on the length of time that the pond has been separated from the ocean, but with ocean sea level change, we know it's that the pond has probably had water in it for about 6,000 years. So we've got a relatively recent colonization event that we're looking at in this particular group. So let me let you in on this detective story. When I showed up in the pond, I, as a seahorse biologist, had this resource available to me. This is the world-known by Sarah Laurie, the Seahorse Guide. The top pictures are Hippocampus reedi. The bottom pictures are Hippocampus erectus. You can notice by looking at them that Hippocampus reedi have longer snouts and slenderer bodies, and Hippocampus erectus have short snouts and fatter bodies. These are a range of randomly selected pond animals. What would you say they are by looking at this? You've got some that have incredibly long noses. This one looks just like a reedi, but then we've got this short nose, heavy, squat-bodied animal. This one looks completely different than both of them, has a very strange head shape. So we saw that there was a lot of variation in their shapes in this pond. And we realized that our first question couldn't be to study their behavior. Now I had another offshoot. We had to actually study their genetics and to figure out what species we were working with. And so to do this, This actually leads me finally to my research questions. There are two main questions we're going to talk about with a little bit of data today. First of all, what species are they? Are they one species, two species, a hybrid, or a new species? That was our first question. And we would expect, based on this predator-free environment and relaxed selection, that we'd expect some drift, some changes in their physical shape from their parent populations, because these are animals that have been isolated in a lake for at least 6,000 years, and thus we'd expect some differences. But we had to actually go through the detective work to actually address this particular question. The second question I'm going to talk a little bit about today in the last few minutes of my talk is to look specifically at their population densities and their sex ratios to start to get to those behavioral questions, and specifically to compare them to what we know worldwide about seahorse populations. Because that's really where the hook is on this story, is do we have a population that represents a higher density population than we see in the world? And again, we would expect that with no predators and with a very constant, calm environment with very little wave action, that we would expect to have increased population densities in this system, and we'd also expect to see, based on our early work, based on our knowledge and our work from Tampa Bay species, we would expect to see that their sex ratios should be even. They're monogamous. We should see even numbers of males and females in these populations. Okay, so how did we do this work? What did we do? Well, for the genetic work, we actually do our genetic work on these animals only by taking little tiny bits of fin from the lower part of their fin here. So fins, when you clip their fins, it's similar to cutting your fingernails. There's no blood. The animals don't flinch. They don't react, other than the fact that you're holding them. They don't like that. And in a couple of weeks, we've re-gone to the same animals we've done fin clips on, and you can see that their fins have healed and actually grown back, because this is something that grows back in these animals. And we can get very reliable DNA from these fin clips. And so underwater in our work, we would find an animal. We would, for the early work, we brought them up to the surface, but we kept them inside bags of water as we worked to make sure they were always underwater as we handled them. And so once we got the fin clip, we processed it, and we sequenced 2 genes from 17 animals. So we sequenced the cytochrome B gene, which is a mitochondrial gene, typically used in seahorses to look to see if there are differences in species. And we also sequenced a nuclear gene, which is used to detect whether there are hybrids or not within seahorses. And we used a bunch of statistics to make these comparisons. We also, though, didn't just look at genetics. We also looked at their morphology, how they're shaped, because that is classically how we determine a species, is looking at their features, their characteristics, how big they are, and how big their body dimensions are. So we took published work from a known paper on a different species, and we used the same measurements that they used. So we looked at things like snout length, head length, so we looked at a bunch of body shape dimensions. And the way that we got this information is, again, all the animals went back after we were done with the work with them. We simply took photos on a slate with basically these little photo things that we can take right underwater with us. We put the animal on it, we take a picture, and then we put them back where they were, and we do all of our biology on those photos, because high-quality photos are where we can tell how much they've reproduced, whether they're male or female, all of those characteristics we can tell from these pictures. Then, to try to figure out where our animals fit relative to Hippocampus reedi and Hippocampus erectus, we relied on collections from my own lab in the work in Tampa Bay for our Hippocampus erectus populations, and we obtained samples from other locations, again, photos of these animals, that we could measure using our same techniques to be able to compare the 2 species and then the Bahamian animals to try to see where they fit. So, and this is just pictures. So this is what our research team, our lab bench looks like. This is the lab bench. It's a rocking kayak where we're working with animals inside. These are mask containers. They work really well for holding animals. And this is our techniques for taking pictures and doing the fin clips all underwater here. This is actually our first undergraduate on the project, and Emily Rose, who's my collaborator throughout it. And so the first thing I want to talk to you about in terms of data is the morphology. And don't even bother to look at the numbers. There's a lot of numbers up there. The key thing you should notice about this is that so what we're seeing here, this is Sweetings Pond. This is the Florida Gulf Coast erectus. So these are the Bahamas animals. These are erectus from Florida. These are erectus from a different published source. This is from Brazil. And this is from another species, Hippocampus patagonicus. And if you look at the ranges, this is the snout length to the head length, which is a ratio, for example, you can see that these animals all overlap. So we can't use traditional measures to compare them because all of the measurements that we can take on seahorses that are usually used to identify what species you're working on overlap across all of these organisms, including ours. The one thing we did notice, which was a really unusual finding, is that when we look at the ratio of what we call how long the trunk is, so the upper part of the body, versus how long the tail is, most seahorses are what we call tail biased. So their tails are very long relative to their bodies. Their bodies are short, their tails are long. What we see in our population, the Bahamian animals, versus the other two populations that we directly measured, our animals have almost an even body to tail. They basically have short little fat tails, which is interesting in conditions where they don't have to fight currents, they don't have to fight tides, they're in a pond that has no current. So that's a possible direction that selection might have taken here. And when we take all of those measurements and we do statistics to look at them, what we can see is that, so these are our Bahamian animals, these are our Gulf erectus animals, and these are our redi. The important take-home message here is none of these overlap. When we look at all of the body dimensions together, our animals look different from the other two possible parent populations. That was exciting. We thought we had a new species. And then the genetic work came back. And so this is just some pictures. We did all of our DNA extractions in the field in our little apartment in the Bahamas. We sent them off to Yale to be able to sequence the DNA. And what we found, dun dun dun, is that they're actually Hippocampus erectus. So our cytochrome B gene, data came back and said that they were Hippocampus erectus. This is the cluster of Hippocampus redi samples at the top when we ran our samples on GenBank. These are the samples of Sweetings pond down here and other Hippocampus erectus, so redi and erectus and our pond animals. They're very clearly part of the erectus clade. And in addition, we've had other folks who have since sequenced our animals, and genome-wide sequencing has confirmed their placement within erectus, and we don't have any evidence of hybrids. So we now know that they're a population of erectus, but they look different from parent erectus, and so it looks like there's a potential there for selection to have acted on this population. So the rest of the work that I'm going to talk about is here. What else did we do with these animals? Well, we wanted to know how dense their populations were to compare them out to the world, out to the larger populations around the world. And so we sampled them across 5 different time points, broken into spring time points and summer time points to look at spring and summer differences. We sampled them along transects so that we knew exactly what area we were sampling to be able to calculate density. And we sampled across the lake to see 5 different zones across that relatively large lake. It's not a great lake, but it's big for the Bahamas, to try to see if there were differences in what was covering the bottom and differences in where we found these seahorses. And again, when we found them, we looked and we took pictures of the seahorses, we took pictures of the bottom, and we scored what habitats were there and how many animals were in each location. And so what do we see in terms of variation? Well, across this mile-long lake, there were 5 distinct zones that we were able to find. Emma's was characterized by mostly Caulerpa, as well as some Laurencia, lots of vegetation in this spot. We saw in the quarry zone, we did see some Caulerpa, but a lot more open areas with oyster reef in this particular habitat, and animals associated with the oyster reef in a lot of different contexts. This south end of the lake, Grouper Cave, was stark, almost no vegetation. We still saw a few seahorses here, but the seahorses we saw were associated with oyster reef. This is all flocculent sediment, like basically light, fluffy sediment. There's very little plant material in that location. Up in the control area, control had only Laurencia algae, so a different kind of algae. And then the place where we started our work, which is known as caves, that's what we call it, caves had a diversity of habitats, basically everything we saw everywhere else, and new stuff, sponges and corals, and a huge diversity of bottom habitats in that particular location. So we saw a huge variation in what was covering the bottom. What did we see in terms of seahorses in these spots? Well, we saw, so this is the density along this axis, so how many animals we're seeing per unit area, per meter squared, and these are those 5 sites I just mentioned. We saw significantly more animals in the north end of the lake, where all the vegetation is, compared to the south end of the lake, where we saw fewer animals in that context. And we saw more animals in the summer than we did, a higher density of animals in the summer than we did in the winter. So there's a seasonal effect and there's a location effect. But again, the question we were addressing here is, how does this compare to densities of seahorses worldwide? That's the orange line. So we already showed here that we see these animals at significantly higher densities than we see seahorses in the rest of the world, which was exciting for us to find. In addition, in terms of sex ratios, so a sex ratio of 50% would be even numbers of males and females. We saw that in most contexts, we did have even numbers of males and females, but in some sites, we actually had more males than we did females. And this pattern in a deeper analysis that we're doing has actually shown to be really consistent across time and across space in newer data that we've added. So we actually see more males in the population, which in wild populations, we tend to see more females. So that's, again, an unusual pattern we're seeing in this population compared to others around the world. And so this is a data table for those others. So this is a list of seahorses that we know worldwide. The key issues we want to see, first of all, this is our hippocampus density, our Bahamian animals. Here we see the maximum density is 0.53 animals per meter squared. The other numbers here, in terms of maximum densities, we saw a few, but this is the average density, and our averages are higher, so an order of magnitude at least higher, if not 2 orders of magnitude higher than everywhere else in the world. And from and the only ones that come close is Hippocampus breviceps that we see in Australia and Hippocampus gutulatus that we see in the Mediterranean. So, in terms of what we're doing next and what we're actually working on right now, we're actually looking at what we're doing as a Mark-Recapture study to try to figure out, so we've got estimates of density from our transects, but we want to have a good estimate for what's the population size of animals in this pond. The typical way we do this with biologists is we have to have a way to mark animals to be able to know if we've seen them before, to identify individuals. Most of the time, the way that marking is done in fish is with some kind of physical tag. So this is actually a picture of us injecting a tiny little bubble of basically, it's a fluorescent dye. It's latex, so it's like balloon material. And we don't inject it into the animal. We just inject it underneath the surface layer of skin. So it doesn't go in them. It just goes like a bubble of skin just right underneath. And it fluoresces, and we can use it to identify individuals. We've used this on a small number of animals in a pond to actually be able to test another question. And the other question is new emerging technology in seahorses, which is using their facial characteristics to identify individuals. Because the patterns of lines and spots have actually been shown to be consistent across the lifespan. And this has been used with another species, Hippocampus gutulatus, to mark individuals and see them over time. So all you need is great pictures of them to be able to put them into a computer and be able to determine whether you've seen them before. So it's an exciting technology we're using to try to get a handle on what's happening in the lake. So in terms of our conclusions, first of all, we know, based morphologically, that these animals look different from the 2 potential parent populations that they could be. But even though they look different, they still have the same genetic structure as Hippocampus erectus. So we have new work going on in our laboratory to figure out what might be driving the genetics of having different tails and the different shapes of these animals. So we're looking into more detail into that right now as one of our questions. We also know that, excuse me, that's the genetics. And we know that their densities are high compared to what we see out in the rest of the world in terms of seahorses, and their sex ratios have some variation compared to what we see in other populations around the world. But of course, there's always the problems that we see. And the problems in this context that we see are what's on the horizon, not what's happening to these populations right now. First of all, this is a picture of what has happened to Hatchet Bay. So that's that water body that was just south of Sweetings Pond. In the 1920s, they cut a hole in between the pond and the ocean to use this as a port, which at that time made sense. There was a lot of agriculture going on. But the unfortunate part of cutting a hole in the headland here is that it changed the habitat in Hatchet Bay from what we believe was just like Sweetings Pond, because it's so close, to a habitat that it looks just like the ocean now. It's got seagrass, it's got large fish, it's got sharks. So it's a completely different habitat than it was. And in addition, Why this concerns us is because there's discussion of making Sweetings Pond a marina. So there's currently discussion in the Bahamian government to develop this area and lose these key characteristics of this really unusual habitat. We also have increases in agriculture now on the pond edge. We've got the development of starting of ecotourism in the area, not associated with our project, but people are starting to find this based on photos and social media. And so because of this, we're really concerned. We're very concerned about this particular system because this is what Hatchet Bay, the marina, looks like on the bottom. It's got very low diversity of bottom cover, and it's covered with trash because it's a marina. People dump stuff off their boats in marinas. That's just what happens. So cutting a hole into the headland is going to ruin the characteristics of Sweetings Pond. But we do know in Hatchet Bay that seahorses are actually found in this location. And these animals, for us, are going to be an important part of the detective story to tell, that look at the genetics specifically of Bahamian seahorses to compare what these look like and their genes with what's happening in Sweetings Pond to find out if the pond animals are actually different in their genetics from what we see in the larger Bahamas. So what are we doing next? Well, we have a number of different research studies that are looking at a lot of aspects of these animals. We're trying to understand what they're eating. We're trying to understand where their overall mating behavior. We're trying to look at things like, we're trying to actually engage in community outreach in these animals. But the biggest next step that we're working on is with the Bahamian government, because we're actually working in conjunction with the Bahamas National Trust, and we're working with an international conservation organization, Project Seahorse, to start the process of moving towards protecting this area and creating a new national park in the Bahamas. It's not just the seahorses and the octopus and the beautiful location that we're trying to protect. We're also trying to protect the 2 miles of cave system that's one of the most unique resources, certainly in the Bahamas. It's a very unusual cave system. There's 2 miles of underground caves, only some of it that's been explored, on the site that has both cultural as well as geologic significance. So these 2 things right in the same site mean that it's a great location to be able to create a national park and catch it before it begins to be degraded by these other pressures working on it. And with that, I'd like to thank my collaborators. There's a lot of them. These are my funding sources. So I'm working with Emily Rose, Jocelyn Curtis-Quick, Adam Jones, and Rebecca Waggett across a range of institutions. And I've got a lot of help in terms of local support. We're working very strongly within country to try to support this emerging process of developing a national park and getting the data to be able to do that. And of course, my undergraduates, which are my scientific babies that we've been raising in this project for the last few years now. I've been fortunate to get a lot of students involved in this project and use this as a great teaching model throughout. And that said, questions?